One of the student, I don't know, I forgot his name, I don't remember his name, and he's from which section of 8th, I don't know. He's told message me that he has not received any notes. Not he, we have not received. He has communicated as we have not received any notes of first and second chapter, that is crop production and microorganism, friend and foe. I have not received any notes. So in the beginning of this, I have given the notes of those two chapters. Complete notes are there. So if anybody have not received the notes, copy it from this and write it in your notebook and make it ready. All these two chapters notes I have given here. You can copy this from this. So we will move to the other things from the cell, the basic structural and fundamental unit. So the first thing, so when we did yesterday, when we had this last week, when we discussed about the structure of a plasma cell, plant cell and animal cell, prokaryotic, eukaryotic, we said that cell membranes are there, cell walls are there, so they have particular function. So we have to study little bit about those things, not in much detail. We don't have much detail in 8th standard. We can study little bit. This also can be considered as a note. If you copy this in your notebook, this is sufficient. Only thing is that the questions are not framed, the statements and everything are correct. Then go back to the back side of the textbook. If there is anything is not there or not explained from this book, after reading the textbook content and the backside, then you can reply to me because you are in 8th standard and our new education policies, some changes has come. So they will start, government will start input those things in the coming years when we don't know much updates and other things we don't know. So please have a habit of reading the textbook. First read the textbook, understand the between the lines, everything, then follow the notes. Follow the notes. So I am not against the notes, but read the textbook. It is very, very important, since the rules are changing. Because you know that in higher education, when we are going, earlier there was no entrance, nothing, only for few like MBBS, engineering, or some, if you want some government seats, then only there were some exams. Now government has introduced for all the courses, BSc, MSc, this, that, for everything they have put entrance examination. So reading is very, very important. The basic things you have to know. So please read. If you have any doubt, comment below. Very important. So go through the textbook first. Consider this as a note. Read the textbook. Go through the back. Solve all the questions by self. Don't wait for the teacher to give. Try to solve it. If anything you can't do, then come back to the teacher and ask what is it. Then do. That will improve, or it will make you to improve your knowledge, improve your ability to understand, reading skills. Many things will come which is further important for your class. Okay, then we'll move to the first one, that is a cell membrane. Cell membrane, which serves as the outer boundary. That means this is the boundary of a cell. It divides a cell externally and internally. So this makes as the outer membrane and make as a boundary. So in plant, apart from after the cell membrane, towards outside there will be having one more layer known as the cell wall, and animals there is no cell wall. And what is the importance of the cell wall? Since a cell wall is the boundary of a cell, so cell wall maintains or controls everything. What should come in, what should go out, when it has to come in, when it has to go out. All the controls are done by the cell membrane. So cell membrane structure is very, very important. I have now forgot to give here. The cell membrane structure is given by a person known as two scientists together known as Singer and Nicholson. They have given the modern cell membrane structure which I have given over here. So you can see many antenna like structures, all of them have some important. The cell membrane are the main structures which are helping to survive from the cold, which are helping from survive from the wind, summer, and these are helping the cell without bursting when it shrink or expand on the basis of amount of solvent present inside that. So cell membrane do all the things. And next comes the cytoplasm. Cytoplasm is a jelly like substance that is present in between the nucleus and the cell membrane. In between the nucleus and the cell membrane, that particular jelly like substance that is present inside, it is known as the cytoplasm. Inside the cytoplasm we have the cell organelles embedded in it. Cell organelles means little organs, that means they also have its own function, they do some functions. And in plants, as I discussed like that, they are having cell wall which gives the plant very rigidity. So that means whenever the plants when they moves in wind and etc, they will give very strength, apart with some other tissues plays a role. And next one we will come to the cell organelle. The first we will go from inside to out. So the first, the cell organelle that is present is nucleus. Nucleus is considered as, is called as the brain of a cell. That means if the nucleus is not there, then cell don't have control. And nucleus is having its own membrane. That means the nucleus itself is covered with a membrane which is called as a nuclear membrane. And inside the nucleus we are having the genetic material, which is, we call it as DNA. It is not found every time, it will be in a thread like structure when we see, so we call it as chromatin. And when it comes for cell division, when the cell want to divide, that time this chromatin and it contents to form chromosome, inside which we are having the DNA. And inside this nucleus there is very highly condensed area which we call it as nucleolus. They are responsible for the synthesis of RNA, ribose nucleic acid, which helps in the which is considered as a messenger RNA, which is considered as a messengers from in between DNA to protein. So this nucleolus is there, and there it is very condensed. So this is the main structure of the nucleus. And the nucleolus was discovered by a person known as Fentona. And the nucleus was first structure was given by Robert Brown. The scientist names are important. And the chromosomes are the thread-like structures which is present on that. And we have DNA. You don't want to study the structure of DNA. I copied all this from Byju's. So you don't want to study the structure of DNA, but you must understand that in nucleus we have something which have the capacity to control information. That information is stored in the form of DNA. And the structure of DNA was given by a scientist known as Watson and Crick. And the chromosome structure was given by Walter Fleming. So these names you must be there, and you must understand that these are present. You don't have to study the structure in 8th standard, that you will study in higher classes. Then with support to the nucleus we are having endoplasmic reticulum. You can see that because these nucleus are having pores, and on that pores we are having some reticulated structure. It is also a membranous organelle and found in eukaryotes, and it is attached to the nucleus. And this cell endoplasmic reticulum was first named by Keith Porter in 1953. These names are important. And on this endoplasmic reticulum we can see some bead-like structures. So according to the presence and absence of bead-like structure, this endoplasmic reticulum is further divided. So if the bead-like structures are present, it is called as the rough endoplasmic reticulum. If the bead-like structures are absent, then it is called as the smooth endoplasmic reticulum. So like that they had divided. And this rough endoplasmic reticulum is responsible for the synthesis of proteins. We will discuss that, what are proteins. And the smooth endoplasmic reticulum are responsible for the synthesis of lipids. And next one, that is called as a Golgi complex or Golgi apparatus. This Golgi apparatus is also a membrane bound organelle. So this Golgi apparatus is the main area which is formed from endoplasmic reticulum, and this Golgi apparatus is the main area where the packing of proteins takes place. Because the message, for example, when we are writing a message, we write on letter and fold it and give. Whatever we are written in that alphabets is the message to one person. Same way inside the cell, protein is the message. If the protein has not reached to the particular part of the cell don't understand what to do. So these proteins are properly folded, packed, and tag labeled to where it has to, like how this Amazon and Flipkarts are delivering the items. So this has to go to Delhi in this particular area. So they are all, everything is labeled same way. Those type of functions are done inside the Golgi apparatus. And this Golgi apparatus, the name is given by Camillo Golgi, the person who first identified this under the microscope. Next one is known as the mitochondria. The person who discovered mitochondria, his name is Kolliker. And then it was Altman coined bioblast. Bio means life giving, that means because of the production of energy. And the term mitochondria was for the modern era, it was given by Carl Benda. Mitochondria is a double membrane cell organelle, which is called as the powerhouse of the cell. Why it is called as powerhouse of the cell? Because it synthesizes ATP, energy. If ATP, like food, as we take food, it will be converted into glucose, and glucose will be transported. Same way, that glucose will be converted into ATP, and that ATP will be used for the functioning of the cell. And this mitochondria is autonomous in nature, that because in nucleus we are having DNA which carries a message. That is why we call it as a control center. Same way, there are few amount of genetic material is present inside the mitochondria, that is why it is called as autonomous in nature, because some of the functions mitochondria can do by itself. They don't want to wait the message from the nucleus. That is why they are called as autonomous. So these are the main things. And next comes the vacuoles. Vacuoles means the empty space that is present. So, and mostly these vacuoles are seen in plant cell having big vacuoles which can be store food, or which can store some other for example chlorophyll or some other things can be stored according to the need and according to the plant. So, and these vacuoles were first discovered by Anton van Leeuwenhoek. Anton van Leeuwenhoek is the first person to observe this under the microscope. And lysosomes are discovered by Christian Duve, and the function of this lysosome is because they digest the cell. Means if the cell is not functioning properly, these lysosomes will secrete some chemical whose pH is very very low compared to the cytoplasm, so they will kill the cell. So these lysosomes can also help in controlling cancer, but not in a full-fledged way every time. Sometimes yes, sometimes no, sometimes they can't control. So these my lysosomes are responsible to replace the damaged cell or the dead cell that is present inside the body. So these are the main cell organelles we are discussed today. So you can write this in your notebook, consider it as a note, and go through the back of the textbook MCQs, fill in the blanks, everything. And if there is any doubt, please comment back. Thank you.